Welcome back to the deep dive. So, when you hear the word cancer, you probably think about, established tumors, treatment, that sort of thing.
Right, the later stages.
Exactly. But today, we're doing something a little different. We are jumping into a frontier that's, well, it's often overlooked, but absolutely crucial, the pre-cancer state.
That's the real window of opportunity.
It is. It's that period where tissues are altered at the molecular level before they turn fully malignant. And at the center of this opportunity is a biological process called cellular senescence.
Hmm.
Now, we usually talk about senescence as like cellular retirement. The cell gets a gold watch and stops dividing. But here's the paradox we really need to unpack.
The double-edged sword.
The double-edged sword.
Uh-huh.
Our sources show it's both a powerful defense mechanism and, if you leave it unchecked, it can become a catastrophic liability.
Exactly.
So our mission today is to really get into that duality. How can this one process be a crucial barrier against tumors one minute and then an active promoter the next?
And that paradox is everything when it comes to prevention. So we're going to map out the entire life cycle of senescence for you, from its initial protective role to its persistent chronic state.
And the key mechanism driving that shift
Is the senescence-associated secretory phenotype, or SASP. We'll get into how it actively remodels that local environment, what we call the pre-cancer tissue microenvironment, or pre-TME. This isn't just about aging, it's a really active process.
Let's dive in.
So let's start with a clear definition. What is senescence, really? It's a stable, and this is the critical part, a largely irreversible exit from the cell cycle.
Irreversible, so it's not just taking a break.
No, not at all. That's quiescence. This is permanent. It's usually triggered by some kind of stress or damage signal. Think, DNA damage overload or the protective caps on our chromosomes, the telomeres, getting too short.
Okay, so how do we, in the lab, distinguish a truly retired senescent cell from one that's just, quiescent taking a nap?
That's a great question, and the distinction is vital. Senescent cells have a very distinct biochemical signature. First, they crank up their internal recycling systems, the lysosomes. They get bigger, more active.
And you can see that.
You can actually stain for it. We use something called senescence-associated beta-galactosidase, or SA-GAL. It's like a blue chemical flag that says, I'm retired.
Okay, what else?
Second, they slam on the growth brakes. They do this by upregulating powerful cell cycle inhibitor proteins. The two big names you'll hear are P16 and P21.
So if you see high levels of P16 and P21, that's a big red flag for senescence.
It's a huge sign the cells are arrested. But here's the paradox you mentioned. They've stopped dividing, but they are anything but quiet.
They're still active?
They are incredibly active metabolically and transcriptionally. They don't just sit there. They start secreting this complex cocktail of inflammatory signals, growth factors, enzymes.
And that's the SAP.
That is the SASP.
And initially that signal, that SASP, is actually a good thing. It's the cell hitting the emergency brake and then what? Calling for help.
That's the perfect analogy. This early or transient senescence is a vital defense mechanism. It works in two ways, through what we call senescence surveillance.
Walk us through this.
Okay, so the first is intrinsic. Just the stable growth arrests itself. The damaged cell is neutralized. It can't divide and become a tumor. Simple.
Right.
The second is extrinsic, and this is often more powerful. It's the immune-mediated clearance.
The call for help.
Exactly. The senescent cell basically puts up a giant flare for the immune system. It displays special markers, and crucially, it secretes these alarm molecules called damage-associated molecular patterns, or DAMPs.
DAMPs, like a siren.
Think of it like a high-pitched siren.
Yeah.
Molecules like ATP and calreticulin, they scream, danger, and alert everyone from our innate immune cells like NK cells and macrophages to our adaptive forces, the T cells.
So the cell stops growing, puts up a sign saying, damaged goods here, and calls the immune cleanup crew. That's pretty elegant.
It's remarkably efficient when it works. We've seen in studies, for instance, with liver cells, that this cleanup is heavily dependent on our CD4 plus T cells. If that process works, the problem is eliminated. Gone.
Okay, let's pause and connect the dots for a second.
Yeah.
So right now, senescence sounds like the ultimate cellular insurance policy. It's a shield, it stops growth, calls the immune system.
And ensures a prompt cleanup.
Right. And we know it's marked by things like P16, and it produces this SAP signal. But this entire system, it seems to hinge on one thing.
Which is?
That the cleanup crew actually arrives on time.
And that is the exact point of failure. What happens when that crew is delayed or they're overwhelmed or the cell itself is hard to get rid of?
The protective shield rusts.
It rusts and its function flips entirely.
And that's the transition to the dark side.
That's it. When transient senescence isn't resolved, it becomes chronic senescence. And this happens a lot with aging, or if the immune system is weak, the senescent cells start to accumulate.
And their persistence is the problem.
Their persistence is no longer protective. It's actively driving the first steps of tumor formation.
So it's not the retired cell itself, it's the fact that it never shuts up. Its sustained chronic SASP is now toxic.
Precisely. The SASP, which was a helpful alarm bell, now floods the local environment. It transforms it into a pro-inflammatory, pro-tumorigenic precancer tissue microenvironment. It's not an alarm anymore. It's an oppressive smog of growth signals.
You mentioned this is a feed-forward loop. How does that work? I mean, what's driving the cell to keep pumping out this toxic SASP?
This is one of the most fascinating molecular details. The loop is sustained in part because the cell accidentally sounds its own internal alarm.
What do you mean?
Well, as the old senescent cell tries to manage its damaged DNA, sometimes fragments of that DNA, we call them cytoplasmic chromatin fragments, or CCFs, leak out of the nucleus into the main cell body.
Wait, so the cell sees its own broken trash as a foreign invader?
That's exactly it. The cell surveillance system sees this leaked DNA and panics. It thinks it's a virus, so it activates a pathway called the CGA sting pathway.
Which is designed to fight viruses.
Right. It's designed to launch a massive inflammatory response. But here, because it's being triggered by the cell's own trash, it acts like a relentless internal fire alarm that never turns off.
Forcing the cell to constantly pump out the toxic SASP.
And making the whole environment fertile ground for a tumor.
And what's crazy is that this is a tiny fraction of cells dictating the fate of the entire tissue.
It's a massive effect from a small population. Even if senescent cells are less than, say, 10% of the tissue, the biological influence of that SCSP is immense. It chemically remodels everything.
Okay, so we've established the paradox for the epithelial cells. Good if they're cleared quickly, disastrous if they hang around.
Right. But we need to move beyond this epithelial-centric view. The problem is bigger than that.
Because a tissue is an entire community of cells. You have the structural support, the immune responders.
Exactly. We have to widen the lens because when senescence hits the stromal and immune compartments, it's almost universally detrimental.
All right, so let's talk about that supporting cast. The fibroblasts. These are the structural engineers of our tissues. What happens when they retire and turn on the SASP machine?
Senescent fibroblasts are active tumor promoters. They aggressively remodel the environment in two ways. There's the soluble SAP, but they also restructure the physical scaffolding of the tissue, the extracellular matrix, or ECM.
And that new ECM is different.
It's much more permissive, much more potent for tumor growth than the ECM from a healthy fibroblast.
Wow.
And we have specific examples. In the colon, when senescent fibroblasts accumulate, they drive this tumor-promoting environment by pumping out a factor called GDF-15.
GDF-15, what does that do?
It's like throwing premium fertilizer on nearby premalignant cells. It activates all these aggressive growth pathways, enhancing their ability to proliferate, to migrate, to invade.
And they don't just help the bad guys, they actively sabotage the cleanup crew.
They do. They suppress the immune response. They secrete IL-6, which calls in immunosuppressive cells. But listen to this mechanism, it's amazing. They put a molecule on their surface called HLA-E. This HLA-E then engages a receptor, NKG2A, on our most important immune fighters, the NK cells, and CD8 plus T cells.
So the senescent fibroblast basically flashes an invisible, like a do not engage signal to the immune system.
That's a perfect way to put it. It tells the T cells to stand down, which cripples the very surveillance needed to clear precancerous cells.
Unbelievable. Now let's address the elephant in the room. Senescence in the immune cells themselves. This is different from the overall aging of the immune system, right?
The crucial distinction. We're not talking about immunosenescence, which is, the general age-related decline of the whole system.
Okay.
We're talking about specific subsets of immune cells right there in the local tissue, becoming senescent themselves.
And you see this in T cells.
We see it clearly in T cells. These P16 plus senescent T cell subsets increase with age and with things like obesity. And these cells, they show exhaustion markers.
So the cleanup crew itself is tired.
The cleanup crew is compromised. Instead of doing their specific job of killing bad cells, they just start pumping out more SASP, making the inflammation even worse.
And this happens in other immune cells too. Macrophages, dendritic cells.
Yes, the senescence of these myeloid cells just pushes the whole environment further toward a pro-tumor state. They secrete their own immunosuppressive SASP. It's a compounding problem. The defense is turning on itself.
So if the problem is this persistence and the toxic output, the solution has to be intervention.
Right, and our sources outline these cynotherapeutics as a critical strategy for what they call precision interception.
They fall into two main categories, right?
Two very clean categories. First, you have the drugs that aim to eliminate the senescent cells entirely. These are the senolytics.
How do they target just the senescent ones?
They exploit a unique weakness.
Mm-hmm.
Because senescent cells are under so much internal stress, they become completely dependent on certain anti-death proteins, like the BCL2 family, just to stay alive.
A survival addiction.
A survival addiction. So drugs like navitoclax, or the combination of dasatinib and quercetin, target that addiction and selectively push them into cell death.
That sounds like the perfect solution, but you mentioned there's a critical caveat.
The caveat is huge. We have to be really careful. While we want to get rid of these bad chronic senescent cells, senescence can be good and protective in other tissues.
Like in wound healing.
Exactly. So if you give a systemic senolytic for prevention, you have to be sure you're not wiping out the good cells needed for vital repair functions elsewhere. It's a major safety consideration.
Which brings us to the second category. Leaving the cell in place, but just shutting down its toxic output.
These are the xenomorphics. They don't kill the cell, they just modulate the SASP. They turn down the volume. They usually target the key regulatory pathways like NFPB or the famous mTOR pathway.
And this includes drugs people might have heard of, like metformin or rapamycin.
It does, and rapamycin is a really fascinating example. Formulations like encapsulated dropamycin or ERPA are showing real promise.
In high-risk groups.
In high-risk groups, like people with familial adenomatous polyposis or FAP, where colon cancer is basically a certainty. By shutting down the SASP, you might reduce that toxic environment and maybe even enhance what's left of the immune surveillance.
And finally, the intervention that's most accessible for all of us listening, lifestyle. How do diet and exercise directly connect to all this complex cellular machinery?
This is where we have the most immediate agency. We know high-fat, Western-style diets promote senescence and SAP. That's a big reason why obesity is a risk factor for so many cancers.
And on the flip side?
On the flip side, things like endurance exercise or calorie restriction work by changing the cell's internal energy gauge. They increase the ratio of spent energy to charged energy inside the cell.
The AMP-ATP ratio. Explain that in practical terms.
Think of it like your phone's battery indicator. ATP is a full charge, AMP is low battery. When that ratio of low battery to full charge goes up, the cell senses an energy deficit, and that activates a crucial enzyme called AMPK. Activating AMPK is like hitting a reset switch. It can delay or even prevent the onset of this premature detrimental senescence in tissues like the colon.
That is a direct molecular link between going for a run and cellular longevity. The challenge then is precision. Suppressing the bad, chronic SASP while preserving the good, acute functions.
That's the whole game.
So this deep dive really shows that senescence is the quintessential biological paradox. It starts as a shield, but if it sticks around too long, it rusts, creating this toxic pre-TME and recruiting other bad actors.
And looking forward, the biggest hurdle we face is what I'd call the marker problem.
We don't have a good way to measure it.
We lack a universal specific indicator. The markers we have now, P16, P21, that blue stain, they're not specific enough. They overlap with other healthy cell states.
Which means we need a much clearer, higher resolution map of what these cells are actually doing.
Absolutely. The future is integrating multi-omics, spatial transcriptomics, to really understand the sheer heterogeneity of the SASP. It's different depending on the cell type, the trigger, everything.
And for the clinic?
For the clinic, the holy grail is developing reliable systemic indicators. Things we can measure in the blood-circulating SSP proteins, maybe, that reflect the senescence burden in your tissues. That would make population screening a reality.
So the goal is to take all this incredible discovery science and distill it down into simple, cost-effective tools that a doctor can actually use for precision interception.
That's the path forward.
So here's a final thought. Considering that simple things like diet and exercise can directly alter our cellular energy state, that AMP-ATP ratio that regulates senescence, if we can one day precisely monitor our body's senescence burden with a simple blood test, could prevention become less about managing vague risk factors and more about directly fine-tuning our cellular lifespans? What step in this whole complex loop, killing the cells, quieting them, or changing our lifestyle, do you think represents the most promising point of clinical intervention?
A lot to think about.
We'll leave that with you. Thank you for joining us for the deep dive.